Hello, everyone. Welcome back to Drug Diaries, your go to podcast for everything happening in the world of pharmaceuticals and biotech. Today, we are diving into some of the most cutting edge innovations in cancer research. Something that is not only revolutionizing the way we treat this disease, but is fundamentally changing the future of healthcare as we know it. And let me tell you, today's episode is going to be one for the books. We are talking about biomarkers, those incredible biological signatures that help us crack the code on how our bodies and specifically cancers behave. Imagine being able to predict how well a patient will respond to a certain treatment before they even start it, or using the body's own immune system to fight off cancer. This is the world of immuno-oncology, where treatments are becoming smarter, more personalized, and dare I say it, more effective than ever before. Now, to help us break all of this down, I am thrilled, honestly, thrilled to be joined by someone who's right at the forefront of this movement. Our guest today is Daniel Lowther, the Director of Clinical Biomarkers at GSK, one of the biggest players in the global healthcare. Daniel has dedicated his career to advancing how we use biomarkers in oncology, and his work is paving the way for the treatments that are not only saving lives, but completely reshaping what we thought was possible in cancer care. It's an absolute privilege to have Daniel with us today, and I can't wait for you to hear what he has to share. So, whether you are deeply embedded in the world of pharmaceuticals or just curious about how these breakthrough therapies could one day change the lives of people you know and love, you are in the right place. Stay with us because you are about to hear how science fiction is becoming science fact, and how the future of cancer treatment is unfolding right in front of us. So without further ado, let's welcome Daniel Lowther to the show. Welcome to Drug Diaries, Dan. Dan, can you walk us through your career journey from your PhD at Imperial College London to your current role at GSK as Director of Biomarkers?
Yeah, sure. So it's a sort of a winding road. It's probably the simplest way of putting it. So as you say, my PhD was in autoimmunity, so studying multiple sclerosis, how the immune system is attacking its own body and in sort of the brain. That was an amazing time in London. From there, I actually went to Yale in the US. I need to work in a lab on autoimmunity, but this is sort of will become a sort of an example for my career is that it's about sort of taking opportunities when they present themselves. And there was an opportunity to work in brain cancer. And there's, again,
Wow.
fundamentally stuck with T cells, but it's on the other end of the spectrum. So autoimmunity is your body attacking itself, and cancer is
Yes.
yourself not being attacked. So it was a chance to use that spectrum and look at T cells in the context of cancer treatment. And that sort of spawned some massively interesting area and spawned a whole lot of other questions that led to going into some of the immuno-oncology side and trying to understand sort of, why are these cells not functional? From Yale, I moved back to the UK and actually joined biotech. So that's the point where I transferred into industry and joined Adaptimmune, which was a very small biotech at the time, but obviously had great growth and it was a phenomenal place to be, a great opportunity that I was very fortunate to get the timing right to join. And that was in cell therapy. So again, fundamentally keeping those T cells at the forefront of everything, but in a completely different field and trying to make industry work. And from there, it's about making science work. So again, it's one of those sort of themes that's like very personal to me is how do we make these things work? How do we actually go from the last two lines of every paper, which is this
Yeah.
can be used for therapeutic benefit, into, well, go on then, like how? And turning into medicines. And for me at that point, it was all about the data. So I was coming in to try and understand the data. This is my sort of foray into biomarkers. But we needed to be able to process and analyze it. So I took a bit of a gamble and went into and taught myself how to code and get into bioinformatics. And I sort of caught a bit of the proof of concept. I can't profess to being the best coder at all, I'm very much self-taught. But it was enough to sort of show how we could use this. And then that paved the way to bring in people who did this and provide that expertise much better than I can. So I was leading that group within the translational sciences team and trying to understand and use our data and make the most from it, which has led into how we use data and it's sort of fundamental to making the most from it. After a period of time doing that, I went into an IT department. It was all
Right.
about trying to make it work for the wider company. I mean, we can get into, but cell therapy is an amazing field. It's real sort of feels cutting edge, but you're trying to build data from different sources, whether it be the research, the clinical data, the manufacturing that you have to do for every patient, because every patient is unique, and trying to bring all of that data together. So I was looking at the data architecture structures and trying to build in that behind the scenes structure to enable the science to work, to enable everyone to analyze the data, to integrate it all together. And from there, I've come to GSK and I've come back more to the science. So now it's sort of back into making the science work for the patients, trying to make the most of the samples that they providing the best insights we can, and really trying to maximize the impact we can make and make the right decisions we can logically make to develop the best medicines and make the biggest impact for patients.
Wow, that's a very fascinating background and a lot of diverse skill sets and experience that you've got. Dan, I want to be a student here. So could you explain your current work in biomarkers for molecular targeted therapies at GSK in layman terms? What are biomarkers and how are they used in oncology and how are they used in drug development?
Yeah, I sort of try and explain it in a simple way, which is it's all about why. So people will come to us and say, why did this happen? Why did this patient respond? Why didn't this patient respond? And we're trying to provide some answers to that question, to provide the context and to really help people understand and make rational decisions going forward. So a biomarker, as you say, is something that can help understand or predict how a patient will respond. Sometimes they're very simple. Do you have this protein expressed? Is it mutated in the right way for the therapy to work or not? But other times it's much more complicated. So it's something you might not expect. It could be a certain patient population or something in their history that they've been to. And so it's trying to uncover that and trying to make a rational decision. Is this medicine for everybody, or is it going to be working much more beneficially in a subgroup of people,
Right.
rather than giving it to someone who it's never going to work in, for example? But it's also then going a bit deeper and saying, well, how do we provide some scientific information from the clinical results we see? Can we explain what will happen to a patient afterwards?
Right.
That's very important. We don't. Not every patient just has one drug now. It's not a case of having one drug and that's it, and you perfect response. So the chances are through your experience with cancer treatment or anything else, you may go through multiple drugs, and the key is, does one preclude the next one? Does one provide an advantage for the next one? And I'm sure we'll talk about it, but how the immune system plays in this and the role that it fills is you're influencing the immune system all the time. So what you don't want to do is prevent it working really well in the next iteration. So it's providing some information for patients, healthcare professionals, for the general field, and hopefully pushing science along, because this is all at the forefront. If we knew exactly what medicine would work before we went, then we wouldn't be doing the experiments. It would just be already on the shelf.
Right. So it's fascinating because we work out these sort of pathways and algorithms for what works and what doesn't. How does that translate into the real world practice where the oncologist is treating the patient, and how do these pathways or algorithms get embedded in the real life settings?
It's a great question. I mean, ultimately, to keep it very simple, the healthcare is an incredibly regulated industry.
Yes.
Quite rightly, in order to make sure that we are only providing benefit to patients. So the health authorities are the sort of gatekeepers in this, of saying, yes, this is useful, this is providing benefit. And every country has their own, and they follow, and they're very rigorous in what they do. So ultimately, it's up to us to provide proof that is a beneficial step in the process. Is that going to provide benefit? Does that allow you to select the right patients who will respond or not? But you're ultimately going to provide that evidence to the healthcare authorities to get on what would be calling the label, which is sort of the right to sell the drug under those conditions.
Right.
Providing in that, and that gives the guidance for the physicians and clinicians to prescribe in that context to the patients. But you'll also then gather real world data. Well, this whole thing is driven by data. So we're looking for things that make impact and work, and that comes out.
What is immuno-oncology and why is it important?
So the immune system is the best drug we have.
Right.
It's protecting us as we speak. The amount of external insults and influences we have are innumerate. The immuno-oncology is really just kind of utilizing and trying to enable the immune system to fight a cancer. As I'm going back to the sort of start with the balance of in a cancer, it's your own cells. So
Yes.
through your development and as you grow, as you learn, like your body is evolutionary has been told, don't kill yourself.
Right.
Fundamentally, those cells have been removed as you developed. What we need to do is kind of break the shackles, prevent, allow the immune system to go and actually attack these cancerous cells. And there's numerous ways to do it. So without trying to go through the whole field,
Yeah, right.
but trying to utilize the mechanisms that are there, either by sort of taking off the brakes to
Mm-hmm.
enable a sort of an increased T cell response, or retraining. So talk about some cell therapy and some targeted therapies are actually about kind of re-highlighting the tumor. There are some markers that we maybe have selected against in our development.
Right.
We don't recognize them anymore, but they're now upregulated by the cancer again. Some great targets to shine a light on the tumor and say, this is not a good cell. So it's really trying to try and utilize all the mechanisms that we already exist. They're already inherent in every person.
Right.
And the most powerful and in some respects least well understood exactly how it all fits together.
Right.
There are many people that know a lot, and I think we've got a very good handle on most of it. But exactly how it all combines is a challenge.
Sure. So that was very well explained. Let me try and summarize this whole thing from my perspective, and you can tell me if I'm understanding it correctly. In our bodies, we've got antibodies that are constantly scouting for the suspects, and we've got these cancer cells who are almost like wearing a cloak, and they are camouflaging or they are appearing to be normal cells. And then these treatments, they help us break that cloak or remove that cloak from the cancerous cells so that antibodies can identify and immune system can go and attack it.
Yeah, so there's many concepts in here, and
Right.
I speak quite a lot in analogies because
Oh, wonderful.
most of my families aren't scientists. So
Right.
if I ever want to talk about my work, then I need to put it into different terms. So if I give you sort of the example in this situation, it's imagine your army. Like your army is not made up of just one sort of set of troops, right? You've got your artillery, you've got your infantry, you've got your air force, you've got everyone. The antibodies are one of those platoons sort of coming along, so they've got a distinct role to. They're proteins, I think, of little Y-shaped molecules floating around trying to bind things. They're almost like your recon. They'll highlight areas to sort of bring in the cavalry. So.
Right.
And that will be things like your T cells, your macrophages, your NK cells. So it's like all of these other players that all again have their own real sort of niche of where they fit in, how they work together. I'd be remiss again if I didn't mention things like B cells, where they will again be highlighting, identifying, presenting antigens to other cells, trying to say this is something you should go and kill. And so you're right in some respects about a cloak. So if you can think of all of these different ways, it's going to be quite hard for something to escape all of them, hide from all of them. They're going to get caught by someone. So yeah, trying to provide either by preventing the proteins on the surface, which would be your kind of beacons for the antibodies to bind to or your cells to bind to, or just generally hiding in areas where they're excluding T cells.
Yeah.
They're just not picked up. They're like playing hide and seek and doing a really good job of it.
Right.
So it's really that's what piece of immuno-oncology will try and pick out a different way of uncloaking, whether it be unmasking and allowing it just to be on the surface, or just making your immune cells better seekers in this context, allowing them to go through. So one of the best, again, my way of thinking about these to keep is with a sort of an anti-PD-1. That has been one of the biggest improvements in cancer therapy in the past couple of decades. That whole thing will be removing the brake. So usually when a T cell is activated, you don't want it to keep going forever.
Yes.
Otherwise, once the threat has been neutralized, you don't want them anymore. Once you've got rid of the flu, you don't want to keep attacking flu. It's not there anymore. It's an enemy. So T cells have a shelf life. There's a brake that's put on. So as they get turned on, they'll upregulate PD-1 to the surface and act as a natural brake. By putting these blocking antibodies, it sort of stops that brake. And that allows the cell, rather than going and, say, killing 3 cancer cells, maybe it kills 5 or 10. And suddenly you start winning the numbers game because you're actually able to be much more effective with what you've got. Or you can break through barriers because you can, get through that proverbial wall.
Right.
So you can make a much bigger impact. And again, it's in that piece. I do think of it as a bit of a numbers game. How do you reset that balance in the favor of our immune system, which is a much more effective drug than anything we can give.
Can you explain how do these immune cells identify the cancerous cells? That because I believe even the normal cells are wearing that cloak. So is there a way they identify that these are the suspects because they all look the same?
Yeah, I think it comes down to the basic. We've got to go sort of back in time to when you're sort of in the womb and developing and even in the early stages of life, is that for a T cell, it's the patterns. So it's using its T cell receptor, and every T cell will have one, and they are, going to analogies, locks and keys.
Mm-hmm.
So if you imagine them as padlocks, everyone's a lock, right? And a cell is its own key in some respects. And proteins can get chopped up into lots of smaller bits, but
Right.
one will fit another one. And through development, you get rid of all the padlocks that recognize yourself. Keys that are yourself, you just say, I don't want them.
Yeah.
They're going to go and kill me. So you lose those T cells out of your repertoire. So you're left with this sort of middle ground, which is the cells that haven't seen anything, but they don't know it's you. So they're ready for almost anything that's coming. And then the hope is that when a virus or a bacteria or something comes in, it's presenting its key, fits a padlock that you've got, and that T cell gets activated, increases in number, and you can go and kill that. For your own cells, in theory, you are masking them, but you're doing it through a non-recognizable piece. So we can do it a few different ways. We can try and represent that target. Because so some of these proteins that are in our cells are only in when we're really young. So some of the antigens used in cell therapy are things called cancer testes antigens. They're presented in the testes and cancers. So they're not generally available in other cells. Or there can be, cancer cells are aberrant in their growth. So they can upregulate proteins that otherwise wouldn't or would be early in development. So you can use those to sort of provide a target and say, actually, go back and have a look at this one. And you can do it various ways. Cell therapy is engineering the cell and putting it back in. So now you've got your own cells, but retargeting them. Or you can use other antibodies to those to act as the linker between the cell and the cancer cell and the drug. Again, various different ways you can. So even within, you can use bispecific antibodies to bring a T cell along. You can just use a monoclonal antibody to an NK cell or a macrophage. There are various ways of doing it, but the key is how do you identify a cancer cell that is not your cell that you want to keep.
Right. That was wonderfully explained. I really love your analogies, and you made this abstract concept so simple. Talking about the translational science, how do you see the role of bioinformatics evolving in the pharmaceutical research?
Yeah, I think it's another great question. It's one of those where I think if you ask 100 bioinformaticians, you get 100 answers. So for me, I think the era of the single biomarker is all on its way out, if not gone, because we've got an amazing array of very bright people who will spot things very quickly. So when there's lots of people looking for things, you'll find them quite quick. So for bioinformatics, I think the key is turning in from what was a single biomarker, where you've got one thing that predicts one response, to a kind of multimodal model. It's not just going to be one. It's going to be a combination of one or two, three or four. And how do you make the sense of that and use it? Because at some point you end up just predicting single people. So how do you make something that is a balance between a complex model and a meaningful insight for many people that can make a large benefit? So that's where I think it's about trying to change the mindset and how we use the data and the data that we generate from things, and trying to really make the right insights rather than just thinking, I'll find a nice easy A and B.
How do you see the data science and analytics shaping the future of drug discovery and development?
I mean, to me, bioinformatics, data science, analytics are all part of the same field. So I think my answer is probably going to be fairly similar to that. Data science is again, it's a different skill set, and I think it brings a freshness of ideas and an uninhibition to what can be possible. So I think it's going to shape medical science and drugs development massively if we can really take the right data sources and create really insightful models, and
Right.
to me, ultimately to guide the right decisions. I mean, this all comes down to the decisions that we need to make, like which drugs do we take through the development path. It's incredibly
Hmm.
costly to make any drug, to bring any drug to market, costs billions. So
Exactly.
any decision you make at a very early stage is incredibly impactful. So the more confidence we can have in those decisions, the better investments we can make, the higher our rate of success, the more drugs and the better quality of life we can bring to patients. So I think it
Sure.
would be about turning in what is possible into what's actually physically real.
Yeah. Right. Can you tell us about the tumor immunology program you established at Yale University and what were some key findings?
Yeah, it was a very very fun time of my career. It was I feel sort of a unique opportunity, and a lot of this was again very sort of timing is everything in everyone's career. I think you'll find that, and then
Exactly.
this was a great opportunity that was to really work in a slightly different field. The key with that program is all about collaboration, and I'm indebted to my mentor from there and how his foresight to like, oh, we could do this, like opening the number of doors that he opened. The key is then can you run through them? And so the program was an idea. To say let's have a look at T cells in the context of cancer, and where we take it from there. And I built up a very strong collaboration with the hospital. So I spent as much time as they would give me with the nurses, with the oncologists, with the radio-oncologists. I was very fortunate to be able to join the tumor board, which was
Right.
humbling because I could see and like I'm trying to understand like how the cancer care was impactful for patients. Like it's
Hmm.
not just sort of theoretical anymore. This is very real. And what do the surgeons care about, and what kind of decisions are they making, and how can we really try and ask the right questions? Again, the big thing from my mentor at Yale was very much ask the big question. Don't ask the little ones. Ask the big one. Go straight for the jugular, as it were, and just like let's ask the most impactful and meaningful. So yeah, it's really a collaboration that we set up and getting tissue. I mean, tissue is precious. It's the most personal and the most like it's a real precious piece. I felt very humbled like every time we got that, but working with the medical staff to say there is spare tissue here. Could we use it for academic research? So I was able to take a tumour that was an excess tumour that obviously the patient we're taking out surgically and try and extract the immune cells out,
Hmm.
which in itself was a Herculean effort because sometimes we'd only get somewhere in the hundreds of cells. But to look at them and try and understand their function and to then say, well, what would it mean? And it was, again, there's an academic exercise in there because you want to understand why.
Hmm.
But there's also it turned into a very real piece because we partnered with Bristol Myers and we're looking at in part of their early trials with nivolumab in glioblastoma, which is one of the most horrific cancers. So it's very motivating to try and make any kind of impact in that field and try and see what was going on. And can
Hmm.
we learn? Can we find out why these patients are responding or not? Can we understand what's going on? Is it
Yeah.
going to work in that setting? Is it worth going extra? So yeah, it was a time full of questions, and it's an amazing setup over there, and I loved every minute of it. So it was being given the tools, the keys to the Ferrari, and
Yeah.
saying, right, go on then, see how fast you can drive it.
Wow.
So yeah, I was massively indebted to the opportunities there, which I think is then sort of I've been hopefully able to come back to the UK and time into my future work.
Right. And how has your understanding of immunology contributed to your work in targeted therapies?
Yeah, the immune system is key to everything. So we've talked about it a little bit, where it's the best drug you've got. I'll put it in terms of a targeted therapy. It's
Hmm.
the question is what's your drug. So a targeted therapy is just one that has a target. It doesn't tell you really how it works.
Hmm.
So you could you could bring a cell along for the fight, and kind of adoptive cell therapy that's targeted to the tumor. You could use a bispecific antibody which binds to the tumor cell and the T cell to bring them together.
Hmm.
You could use a monoclonal antibody that brings an NK cell in, or through just the killing of the cell by binding it, it could block the signaling of that cell. So really, it's just about highlighting the tumor, saying this is
Yes.
tumor, this is not me. I don't have to use the the systemic therapies. I don't have to kind of use chemotherapy or like systemic chemotherapy where I'm really dosing some pretty horrible drugs throughout the whole body and hope, like with the idea that I kill the cancer as fast as I can to spare the patient as much pain as I can. It's saying, well, can we target it? Can we try and provide a much better quality of life? Can we really hone in on what we're doing?
Right.
Yeah, I'm trying to sort of think of the analogies, but like storming the castle, you only want to get rid of the keep.
Yeah.
Literally lay the whole thing low.
Exactly.
So it's going to go into that. So, but using how you use the immune system and how you complement it and work with it, with the therapies, and is it going to be a friend or a foe? Sometimes the cancer is in the immune system. Some of the hematological malignancies, like
really?
Fundamentally could be a T cell that is growing too fast or a B cell that is growing out of control. So sometimes it's about eliminating them. It's trying to find the one piece,
Right.
and not all of it.
Yeah, it's like you have to choose the right ammunition from your arsenal that would work the best and with minimal collateral damage.
Absolutely. I mean, again, we've got to remember as patients, everyone will know someone that's gone through cancer treatment. It's a horrendous experience. So, anything we can provide, any kind of improvement in quality of life is massive.
Exactly.
It's avoiding that collateral piece is
right. Talking about innovation and challenges, what do you consider to be the most exciting innovations in biomarkers research right now?
I do think it's the fresh ideas coming in from the sort of data science field, from AI/ML.
Right.
It's really going to be about taking data and maximizing it. So how do we provide links that we didn't expect? Allowing the sort of a model and machine learning model and approach to identify links that we wouldn't necessarily think of. Sense check some and go, does that make sense? We still also need to be able to explain it.
Yeah.
Highlighting it, coming up with a hypothesis of saying, well, actually, have you looked at these two things next to each other? Did you think about this subgroup of patients? Did you think this might be how the drug worked? I think that's going to be the biggest change and disruptor to the industry if we can maximize it and if we can use it and learn from it properly. And that all comes down to asking the right questions. Data will give you an answer. Is it a useful answer? And that comes down to how well you define the question you were asking, how well you set up the model with the right assumptions, with the right balances, with the right data sets. Did you generate the right data sets in the first place that will answer that question? So I think that's the biggest innovation, but also the piece where we're trying to find out about right now and trying to get comfortable with.
Right. And what are some of the biggest challenges you've faced in your research career and how did you overcome them?
Yeah, I face challenges all the time. I think the key with all of this is it's not a one-person job. The key is where you have to work together. It's a constant battle to really make impact and provide impact, but you cannot do it alone.
Yes.
So understanding and working with others is always a challenge. I think that the biggest strength of humans is that we can work together and communicate, but it's also the piece that brings a lot of the challenges that come with it, because everyone's got their own desires, everyone's got their own motivations, and everyone's in a different stage of life. So how does everyone get something out of this? How do you work with them? How do you understand what they need, not just what I need? How can you listen? How can you try and find win-win scenarios? And whilst achieving and moving forward and picking them up. So I think the challenge for me is always in that way. Personally, I know I'm trying to be as much self-aware as I can be. I know I tend to jump to the end.
Yes.
I don't know whether it's right. I've got there. People have different processes. Again, I'm never going to say I'm necessarily right. I might not be wrong plenty of times and will be, continue to be. But I still need to be able to explain how I got there. Others walk along the journey too, because if they're right and I'm wrong, I need to know that. I don't want to be taking decisions that take us on the wrong track. So it's providing that space. It's not assuming I've got all the right answers. It's allowing others to really influence me and have as big an impact as they can have. So that's probably the challenge. It's an ongoing piece in every field. So my career's gone through different fields and really listening hard in each one, because everyone brings something different. The IT sphere was a very different experience. Being in a back of house role, the front of house role was fascinating. It's a whole different set of challenges. And I urge anyone to go and take a back of house role for 2 or 3 years. It gives you a very different appreciation of how business works. and what people care about and how change affects things. So, but getting comfortable with that, like it wasn't a comfortable place for me. I'm sort of action orientated and want to move forward. So setting up things in the background, like really trying to bring people along in change was a real challenge.
Right. And that leads us very nicely into the next topic of interdisciplinary collaboration. So, what are the disciplines that you typically work with in your day-to-day research work?
Almost you name it, everyone. I mean, the old sayings, right? It takes a village. Like it
Yeah.
does, if not a town or a city. The, there's a lot of unsung heroes that really make things possible. And again,
Hmm.
they're unsung heroes because you don't necessarily think about them.
Yeah.
It could be through an ops team that are, ensuring that patients are recruited in the right way, that samples are processed in the right way and sent to the right labs. I don't physically see the samples. Since my academic time, I've not had to deal with
All
any
right.
samples
Yeah.
because if we're dealing with global trials, there are sites across the world. How do we provide instruction for those hospitals to collect the right sample? For that nurse or that team of nurses to collect the right samples to ship them to the same right place. And then again, our whole ops team who work with are phenomenal in that and really, again, just underpin it. And you've got all the other functions that go with it. So from facilitating the work to analysing the data, all our analysts, all our clinical folks and colleagues, and who are helping put everything in the context where it'll actually work, what will develop, what is possible, how will people react. Some of the medical affairs teams that are out in the field, they're talking to people, they're understanding like what do people matter with, what do people care about, what's the biggest unmet need, burning scientific question that they want to answer. Like,
Exactly.
that's one of the things I enjoy the most. I love being able to try and make connections between different groups of people. Again, I think it probably speaks to my career and the way I've gone, and that I've not done the same thing for more than kind of 3 years at a time. It's been about that connection. It's been about that interdisciplinary work. It's about learning from others. I had a great mentor back in a research project I did in my undergrad, and he had a lab up in Buffalo, and he said to me quite early on, was like he never, I think of maybe one exception at the time, hired the same person twice. Someone that previously
Right.
worked for him wouldn't, and it wasn't because he didn't like them; he loved them. But it's all about freshness of ideas. It's all about someone coming in from the outside and saying, just sort of cutting straight to the point. Like an outsider view will often just ignore all of the assumptions and say, but that doesn't make sense.
Exactly.
And I think having that diversity of thought, diversity of view, diversity of perspective is massive in the whole business. If we're going to make it work, we can't possibly expect for me or for any one person to have all the answers. So trying to involve everyone in the process rather than even just relying on saying, that's your job, you just do that.
Mm.
No, people are motivated by the why. People want to know why what they're doing matters. I think everyone's got
Yeah.
that right. How we evolve and how we all work together is kind of key to this whole thing.
Absolutely. And from your experience, can you give us an example of a successful collaboration when everything just perfectly came together and fell in the right place?
Yeah, I think it happens all the time. I think it's one of those where everything that works is an example of that. I mean, the immunology program at Yale, I call it a program only because it was so many people.
Mm.
It wasn't just me. I was one part of this thing, and hopefully I played a part in enabling others to be successful too. And working with the hospitals, making that link. I think that kind of collaboration enabled. I mean, again, it enabled me to do my work.
Yes.
So they were enabling me. They didn't need to. They didn't have to. I hope that they wanted to. And however we got to that point, and
Mm.
through chatting, through sort of idea generation, through the idea of again making an impact and trying to make the most out of things, is really trying to get to that kind of collaboration. But again, it would not be possible without, well, all the way back to the patient. The patient has to be willing to provide the tissue. It's really a sort of everyone buying into the same idea, and that really enables the potential, say, development of treatments, or for us to justify a trial, which again may work, or hopefully will work, and then hopefully provide benefit to extra other patients. So I always come back to like drug development is a collaboration right from the start. Patients who enroll in trials are they're so generous. because they're providing sort of deferred benefit that hopefully the treatment will benefit them. But the reality is, like, the big payoff is for people in the future. It's from us learning from these as we go. I think that's yeah, I say incredibly generous of them. So yeah, everything is a collaboration.
What has been the most rewarding aspect of your career so far?
I found so many things rewarding. It's hard to pick a kind of, oh, it's that. I've got something from kind of everything I've done. I love doing different things and bringing the links together. So hopefully it's where I've found things rewarding when I've learned the most. When I've stepped into a new field and gone, I really am uncomfortable here. I need to put the effort in to get comfortable to do. Like, I know I can add value. How do I add value? Where do I so that challenge is incredibly rewarding. Hopefully at some point I've inspired others. I'm a father to a couple of boys, and it's really personal. I get so much pleasure from seeing them grow, and it's the same in the work, in the career environment, of trying to provide others the opportunities that I had, trying to give people confidence to take that step, to help create that opportunity for themselves, to run through that proverbial door, even if it's only just open a tiny piece. I find I get a lot of pleasure and sort of contentment from seeing others' success. That's incredibly rewarding. It's the sort of the rising tide floats all boats kind of piece, and it is. It's like watching others grow and make the most of opportunities is great to see. So I think in this whole piece, it's like, for me, science, it's about trying to make science work. And it's a group effort in this. It's not about one. I mean, I've got my rugby shirts up on the wall. These are sort of training days, and it all comes back to that team aspect.
Yes,
we've all got our roles to play. We all have our sort of our specialisms and our piece. But when the game starts and the balls are on the floor, it's like, you just got to do what's necessary. You're there for your teammate. That's the key. Like, you might not be quite as fast as your opposite number, but you've got your teammate backing you up. Like, you're going to be there on their shoulder. You're going to be sort of all in for them. You've got the collective piece. So that's where any time that things have gone in that direction, they're the most rewarding experiences. Anytime you can share something with someone and go through something, go through a challenge with them, overcome it with someone, or like you got someone to talk about and go, that was hard, wasn't it? Yeah.
Yes.
Like, it's for me anyway. It's more about that kind of piece than necessarily a personal, like, I have to do it.
Absolutely. That's a very rewarding aspect of collaborating and helping someone. Yeah. Now, obviously, the work that you are in is very demanding. How do you maintain a balance between your research work and your personal life?
For me, it's all about priorities. So it's all about doing things that are in line with your values. And if you're acting in line with your values, you have balance. Yes. So everyone's balance is different. What is balance for me will be different to someone else. I've always enjoyed and wanted to prioritize my life outside of my work, but also prioritize the life of the others I share with and their work and other people. Like helping provide the space for my wife's career. And my kids, and wanting to spend time with them and watching them grow and helping them develop and make the most of the opportunities they're given. So that's so important to me that I want to prioritize that. Now, how I prioritize that will be, and then how I approach all the other things in my life. Work being an enormous one, but trying to be efficient, trying to be the most thoughtful and effective with my effort. So when I do it properly. It's not about doing something for twice as long and half the effort. It's like, let's go in, let's get it done. And again, very action orientated, but being that piece. But I never tell anyone that works really long hours that it's wrong. It's all about what they care about. I mean, scientists as a whole, like we love interesting questions. And for a lot of people, like, science is their hobby as well as their work.
Yes.
So if it takes up 15, 16 hours of your day, that's not bad. If that is in line with your values, then you're doing it. There was a piece in a sort of career sort of seminar that from a long time ago my wife told me about, which was that she went to, and it was, rather than representing here's my career, and it was an X-Y graph. Time is on the X-axis. So and usually you might think on that Y it's like seniority or something else, and it wasn't. Yeah. It was happiness, and it was like, oh, really? This is how to judge your career. Like your happiness goes up and down at different times, like, that are not necessarily linked to any one thing in particular. When we were thinking about it, it's like, is it the word happiness or is it kind of contentment? Is it that
Yes.
satisfaction? It could be anything. It's one of those synonyms. But the key is like, that's what you're striving for, to maximize your contentment, your satisfaction. Ultimately, if we group call it happiness, it's not necessarily about just being joyful. It could be the sort of, that deep satisfaction of saying, this was a real challenge and I overcame it. It was really hard, but I did it. And trying to just enjoy the journey. It's all about the journey. If you focus on the goals, you never get there. You'll get to a goal and there'll be a next goal. You're sort of deferring that satisfaction by saying, I'm not satisfied right now because I'm trying to get to this. So the journey you're going on has to be the thing you want to do, and that's everything we do. So that balance is everything's a journey, both at home, at work, in everything.
Very profound and wise advice.
Try my best, but whether I can live it is a challenge.
And what advice would you give to young scientists aspiring to make an impact in the biopharmaceutical industry?
Yeah, I love talking with young scientists. I love trying to provide that piece. I think the key is about making the most of opportunities, but also creating opportunities. So I would always say, talk to as many people as you can. I ended up at Yale because of a conversation that I had at a conference. If I hadn't had the conversation, we wouldn't have got there. If I didn't follow up on the conversation, it would have been a no straight away. Taking opportunities and really sort of finding them, but ultimately it's just following your nose, doing what you find interesting. If you're constantly finding things interesting and doing that, then you're to the previous piece, like you're probably going to be pretty satisfied. You're probably going to enjoy it. It's going to be motivating. Doing the thing that's in line with your values, like that's fundamental to everything. If you're not doing things in line with your values, then you'll be frustrated. You'll be unsatisfied. It will have negative emotions. If you're doing things in line with your values, and the more you can get comfortable with your values, it's a very uncomfortable piece to go, that's what I enjoy, that's what I want, that's what I value. And it will change over time. So you don't have to have a 20-year plan, because the moment you have kids, life changes, perspectives change, priorities change. It's about saying, like, who am I? What do I want? How do I go after that? What's going to be the thing that gives me the most contentment? That's going to be the most motivating for me to do. And if it lines up with those, then I don't think anyone's going to be in a negative thought. It's going to be very empowering, and you don't know where you're going to end up. So but you'll be in a good place.
Exactly. That's a very, very sound piece of advice. And here comes another philosophical question. If you could go back in time, what advice would you give to your younger self at the start of your career?
Yeah, enjoy the journey. Like it's I know for me, the thing I have a hard time with is control. I think I'm not unique in this, but like I want to feel that I sort of got some semblance of control over things and try to, which is one of the most frustrating things. So trying to let go of it a little bit, enjoy the ride, not try and over plan, but just I think it's really about sort of taking that opportunity. Just say, I like to think in the career I've said yes a lot, which is where it's sort of ended up in this sort of non-linear path and looking at things for the benefits they can be. So it's really trying to sort of double down on that and just enjoy the journey. Probably the best advice I can go back to my old self.
Absolutely, that's absolutely perfect. And that brings us to the end of our today's episode, Daniel. I can't thank you enough for joining us today and sharing such valuable insights into the world of biomarkers, immuno-oncology, and the future of cancer therapies. Your ability to break down these complex abstract concepts in a way that not only I, but our entire audience can understand has been truly remarkable. And it's not often that we get to hear firsthand from someone who's working at the forefront of innovation in such a critical field. And I'm sure everyone listening today has gained a much deeper appreciation of how science is evolving to change lives. From your career journey to your pioneering work at GSK, it's clear that your dedication is helping us shape the future of cancer treatment. And for that, we are incredibly grateful. Thank you once again for your time, your expertise, and for helping us navigate the fascinating world of biomarkers and oncology. It's been an absolute pleasure having you with us today. And I know our listeners will walk away from this episode with newfound knowledge and inspiration.
It's been my pleasure. Thank you so much for having me.
Thanks, Dan.